evening, and sir, and respective delegates and participated people in the conference. Today, I am going to present my entitled of Association of Bracken Crabs with Corals from Andaman Nicobar Islands, India. I am Dr. S.Kumaralingam. I was worked in last 10 years in Zoology Survey of India. By tenure time, I was done my doctorate Bracken crabs in Andaman Nicobar Islands. One of the chapter is Association of Bracken crabs with corals from Andaman Nicobar Islands. Introduction. Coral branches provide shelter and protections for the associated crabs. Comparatively, the head-forming corals contain a more symbiotic individuals than the other types of corals. Biodiversity and community structures and morphology of coral hosts are related with the abundance of associated crabs. Coral-associated crabs, including both the anemurans and brachyurans. Comparatively, the brachyurans were highly associated with the coral reefs. A large number of brachyuran crabs are associated with both the dead and live corals. Agropora associated crabs were reported by Sushanji and Nijoma 2002. The symbiotic crabs densely aggregated on host and show little territorial behavior and more frequently between the host individuals. Compared to the coral genus Pocillopora, has higher abundance of the crustacean fauna other than the Agropora. Crab associated with the Pocilloporidae corals were found to be a highly concentrated probably because of they inhabit a unique coast of, and it is easy to absorb the interaction. The present work is preliminary effective exploration the association between live corals and Bracken crabs community. And materials methods. The whole colonies were collected samples randomly from the reef of selected islands of Andaman Nicobar Islands. The colony samples were branching corals of the family Pocilloporidae and Agroporidae. All census were conducted by use scuba apparatus. At the each reefs 10 into 30 in 1 meter belt transect. Helen Milsium 2004 were laid in 1 meters above their parallel to the crust. This was a design to the minimized exposed related difference in corals abundance among the transects. All Pocillopora and Agropora corals within the belt transects were identified to count of the corals colony and sample collection made from the January 2011 to December 2013. In the dead islands of Andaman Nicobar Islands we covered. And it is showing the figure 1 belt transect method. A is a steady size and B at each reef of 10 to 30 meter into 1 belt transect. And C count of the coral colonies. And the D is the absorb of the Corallococcus crabs. And E collected the Agropora colonies. And F coral translated to the laboratory studies. We analyze the crabs and which corals were living out there. We'll be finding. And the result, a total of 329 coral colonies were sampled, and 155 Agroporidae and 179 Pocilloporidae. Among the 12 species symbiotic which belongs to the 3 genera and 2 families from the Andaman Nicobar Islands. The associated crabs of Andaman Nicobar Islands show the highest species among the North Andaman, followed by the Little Andaman and Nicobar group of islands. At table 1, we can see them. The highest percentage of Corallococcus were observed in the Agropora and Pocilloporidae and recorded in the North Andaman 70% and the abundance is 67.21%, followed by the South Andaman and Little Andaman and Nicobar Islands. And here the distribution of coral reef associated crabs in the different studies. Among the 10 islands, we are separate the 5 different groups, and the North Andaman and South Andaman and Little Andaman and Angkor group of islands and Great Nicobar Islands. Among that 12 species have like evenly distributed that 5 particular area and the 10 islands. And in exclusion in that number 3 serial number, Tetraloid nigrogenata and the 6th Trapezius septa. These 2 species are among the present in all the islands. And discussion. The presence of symbiotic association of Bracken crab in specific corals, particularly 2 different coral families. In the table 2 we given. And a total of 12 species belong to 3 genus and 2 families are observed in the symbiotic to 2 major coral families such as Agropora and Pocilloporidae. And significance post-laporal damicorals provide a maximum host specificity to the 5 crab species, particularly for the Trapezidae family. Also, the Acropora formos and Acropora symmetry provide a 3 different types of crab, such as the Tetralidae genus and Quadrilla and Trapezia species. The Tetralidae species of the corals entirely associated with the family of Acroporidae corals, while they are lost the specificity with the Poecilophoridae. The Tetralidae nitrogenous was found in all observed in the Acropora species, while absent in that Poecilophoridae. The table to the presence of symbiotic of associated vacuum crabs in the specific corals. On that the 12 species and Agroporidae, Agropora formosa, Agropora microdesis, Agropora symmetry, Agropora harmonia, Agropora valeta, and Poecilophora damicorals, Poecilophora vericosa, and Xylophora pistillata, and Seriotopora hestin, and Seriotopora stellata. These are among the 10 species among the 12 species presented exclusively on the Agroporidae present the Tetralidae families, and the Poecilophora mainly present the Trapezidae family. Among the recorded species, Tetralidae, Nigrolgent, and Trapezidae are observed in all the steady sites. The maximum percentage of Coryllococcus crabs were observed in the North Andaman, and 70% percentage-wise and 67-21 for abundance, while the minimum of Great Nicobar 50% and abundance 58% of the corals of Acropoda and Poecilopoda respectively. I believe in 1976 state that the different proportions of Coryllococcus and non-Coryllococcus species are found in the based on the environmental variability due to the physical and chemical parameters are affecting the Coryllococcus species, while Coryllophilus was escaped from the effects. Excluding the Coryllococcus mean they depend on only the coral living organism of crabs like a cryptophanas. Non-Coryllophilus means beyond that like animal crabs and Coryllophilus mean that the large crabs like Xanthidae crabs. They are mainly supported on that coral living organisms, and that sometimes that coral species the Acrontheta spensi sea stork they're going to bite that corals. That time the crab will be disturbed into the coronus like that Echinoderms groups. So the final conclusion is the systematic analysis of species abundance of Coryllococcus or non-Coryllococcus crabs or is needed because they are directly or indirectly help to that coral reef environmentals. So we can conserve the coral reef environmental as well as we can conserve the cryptofauna species also. And I acknowledge to my own institution like Zoology of India will provide the opportunities and Port Blair campus and my headquarters in Calcutta and the Ministry of Animal Forestry and Climate Change. They given the fund. That time I done my doctorate and even I worked towards done this particular paper work. I was done that one. And live DNA they only provide a fund for this conference 20%. And finally, thanks to that Asian Conference Science and Technology and Medicines, they are giving a good opportunity to me. I will present it in front of you audience. Thank you.